So we covered last chapter, chapter 6, the formation of sugars. Capturing the sunlight, the energy in the sunlight, and converting it to chemical bonds in sugar. So now the producers have trapped sunlight energy into a chemical bond that can be used now at any time. It's a very stable chemical bond, as we talked about. So now this can be stored. It can be stored as a simple sugar, but most of the time it's going to be stored as a complex carbohydrate, putting those sugars together to form big carbohydrates, starches in plants. And so, and remember, plants also will reorganize those sugars to form cellulose, which will be used to build the plant body, and then later can be used to form woody tissue. And so all of that is coming out of the products of photosynthesis, out of sugars. So again, sugars are all important. So this is chapter 7. We just covered photosynthetic organisms capture solar energy. It's stored now in sugars, so it's there, it's good to go. Energy stored in sugars must be converted into molecular energy in the form of ATP. So took sunlight, made it into ADP and ATP and NADPH, and then took that through the Calvin-Benson cycle to form sugar. Now it's set, it's stored, it's bonded. But when even plants need energy, they will break apart those sugars and form ATP again. And so ATP is being made and recycled as we go through this. Most eukaryotes, that's cells that have nucleus, so that's plants and animals and fungi. Some bacteria break down carbohydrates via aerobic respiration. Aerobic means oxygen. Respiration. Now, why is the breakdown of sugars called respiration? Because we think of respiration as breathing. Well, a big part of breaking down sugars is gas exchange. For you have to keep breathing to get oxygen constantly. What happens if you stop bringing in oxygen? Well, you pass out, or you could die if you don't have oxygen. Why do you need oxygen constantly? Well, to live, I realize that, but why do you need oxygen? Think about that. What does oxygen do once it gets inside of your body? Yeah, your bloodstream carries it around your body, your heart pumps so that you get oxygen everywhere. Every cell needs oxygen. Why? You have to have oxygen to break down sugar. Remember when sugars were made, you needed carbon dioxide and water and sunlight to make the sugar. This is now, it's the exact same reaction, just backwards. We got sugar and oxygen. Now we're going to break that apart, and what's going to be our products? Carbon dioxide and carbon, I'm sorry. We need an oxygen. So all right, welcome to chapter 7. Chapter 6, we just finished photosynthesis, where plants take the energy of sunlight and convert it to ATP and NADPH, but those aren't stable. So then it takes it through that Calvin-Benson cycle and makes sugars, which are very stable. Now, plants, when they need energy, they typically don't just use the ATP from the chloroplasts. They're going to use the ATP as they break down sugars. So remember that the production of sugars is so important because this is our energy. Now, if we're not going to be using that sugar quickly, what plants do is that they make much more glucose than they need. And remember, they start putting it together into a big giant molecule, a polysaccharide, a complex carbohydrate called starch. So then when the plant needs sugar, it can just go to that starch and start breaking off the individual glucose molecules. We do the same thing as we eat. We don't make sugar, but when we need energy, we eat a lot of carbohydrates. We're getting in plant material potatoes and flours and breads and pastas. So, but as we digest that, we are taking that sugar and is breaking them off, again, off the starches, so that we can use that for energy. Now, when we get too much sugar, we don't form starch. We start putting that together. Remember when we went through the molecules of life into glycogen, which will be transformed into a fat for energy storage. So this chapter 7 is, well, now we've got the energy unit stored, but it's not going to do us any good stored. We've got to get it broken up out and get ATP. So photosynthetic organisms capture solar energy stored in sugars. The energy stored in sugars must be converted back to molecular energy in the form of ATP for use for energy requiring reactions. Most eukaryotes, that's organisms that have a nucleus, remember that, so that is plants and animals and fungi, protists. So most, and some bacteria break down carbohydrates via aerobic respiration. So that oxygen has to be present to break it down. So remember we talked about why are we breathing in oxygen? So that we can break down sugars and get the energy that we need. So there's two main metabolic pathways to break down sugars. By far the best and the one we use most, aerobic. Breaking down sugars with oxygen. Anaerobic, on the other hand, is with the absence of oxygen. They're used by prokaryotes, remember the bacteria and archaeans, and some protists in anaerobic habitats, like at the bottom of a pond, down in that silty area of the pond where it smells like sewage. There's no oxygen down there, so bacteria and protists can live down there, but they don't have oxygen, so they have to break down sugars differently. And that is called anaerobic respiration. In modern eukaryotic cells, most of the aerobic respiration pathway takes place inside of the mitochondria. Remember from chapter 4, inside the cell. The mitochondria was the powerhouse of the cell. It makes the ATP. That's where the sugars are broken down if oxygen is present. Like chloroplasts, the mitochondria have an internal folded membrane. Now what's going to be neat about this chapter is if you have learned chapter 6 really well, chapter 7 is going to be a breeze because it's pretty much the same process but in reverse until we get to, remember, as you know, we did the electron transfer chains in the thylakoid membrane to pull hydrogen ions in. That's exactly what's taking place in the mitochondria. So the mitochondria is acting like the thylakoid membranes in our cells. But instead of having chloroplasts that will absorb sunlight to get the energy, we're going to get the energy from these breaking apart the sugar bond, using that energy to drive production of ATP. All right, so electron transfer chains in the membranes and the mitochondria set up that hydrogen ion gradients. We're pulling in hydrogen ions inside, just like in the light-dependent reaction, to power ATP synthesis. Again, what's going to be happening in the mitochondria? Exactly the same as the light-dependent reaction. Pulling in hydrogen ions, they can't get out. There's only one place they can get out. Same enzyme, ATP synthase. So instead of sunlight being used to pull the hydrogen ions in, we're using the energy trapped in the sugar bonds to pull hydrogen ions through that membrane system. At the end of these chains, electrons are transferred to oxygen molecules. So photoautotrophs, those who use light energy to make sugars, make ATP during photosynthesis and use it to make glucose and other simple carbohydrates. Most organisms, including photoautotrophs, make ATP by breaking down glucose and other organic compounds. Even plants break down sugars for energy. They don't use the energy made in the light-dependent reaction for energy. They don't use that ATP and ADPH. They only use that to make sugar. To get energy, they're breaking apart the sugars that they've made also. So here's just the two chapters. Here's chapter 6, photosynthesis, carbon dioxide and water. And here is what's the products? Glucose and oxygen. Now for this chapter, we got glucose. We breathe in oxygen, and what is the products? Carbon dioxide and water. And so there we go. There are 3 stages of aerobic respiration. And you'll need to know each of these stages on the test, unfortunately. The first is glycolysis. We'll cover that in detail here in a minute. Then acetyl-CoA formation and Krebs cycles. We got another cycle, like the Calvin-Benson cycle. And then the electron transfer phosphorylation, going down the steps, pulling in the hydrogen ions. Here's our formula. C6H12O6, glucose, plus oxygen. Now we're going this way. Gives us carbon dioxide and water. Remember last chapter, it was just flipped. Carbon dioxide plus water gives us glucose and oxygen. Now it's just the exact opposite. We're going backwards now. The coenzymes, so we have 2 coenzymes. They're a little bit different. This one, instead of NADP to make NADPH, we're just using NAD, adding a hydrogen to it. And then there's an FADH2, which carries electrons and hydrogen. All right. Here's a breakdown of this process. This is aerobic respiration. Here's the glycolysis, the first step. It takes 2 ATP to get it started. Remember we talked about activation energy? That you got to add just a little bit of energy to push that boulder over the hump. Well, there's enzymes here that make it very simple to start. It only takes 2 ATP to get this started. So this is, photosynthesis was an endergonic reaction where energy was taken and stored. This is an exergonic. But you still have to invest just a little bit of energy to get this started. So this is the activation energy. It's been reduced by enzymes. But it takes 2, and you'll see out of glycolysis we get 4 out. So how many ATP did we gain? Just 2. It costs 2, we get 4, we gain 2 ATP. Then we go to the second stage, which is the acetyl-CoA formation and the Krebs cycle, which is combined here together. And you're going to see that what are we going to get out of this? Well, we're going to release a bunch of carbon dioxide, and we're going to get 2 ATP. That's still not very much energy coming out of a sugar. It's the third stage that is the big energy payoff, the electron transfer phosphorylation. Going down the steps, getting the electron excited, going down the steps, pulling in hydrogen ions, and as they go out of ATP synthase, they will make 32 ATP. So you see why oxygen is absolutely critical. This part can take place without oxygen. This part can't. There's some organisms, bacteria and archaeans, that can survive with just this because they don't have mitochondria. They don't use oxygen to break down. So for every sugar that a bacteria breaks apart, he's only getting a net 2 gain of ATP. But they're small enough, they're single cells, they can survive with that. You have 34 trillion cells. You can't survive like that. So you've got to have that constant input of oxygen so you can carry out the Krebs cycle and then lead into the electron transfer phosphorylation. Comparison of aerobic respiration and fermentation. What is fermentation? That is anaerobic. I haven't told you that yet, but they put it up here on the slide. Without oxygen, there's only the net gain of 2 ATP, what we just talked about. Here we have a net gain of 36. So for every sugar, on average, there's different parts of the body that break this down differently, but on average it's 36 ATP per sugar. If there's no oxygen, then it's only 2 in fermentation. And then it gives you another final product. What's the final product? In plants, it's ethyl alcohol, moonshine. In animals, it's lactate or lactic acid, which gives us that burn in our side as we're running and we're not getting enough oxygen in our system. So that's the breakdown of the 2 different ways of breaking down sugars and what you get out of it. All right, so aerobic respiration and fermentation both begin with glycolysis, which converts 1 molecule of glucose into 2 molecules of pyruvate. So glucose has 6 carbons, Pyruvate has 3. So basically, in glycolysis, you're just taking a sugar and breaking it in half, and that's your products there. After glycolysis, the 2 pathways diverge. Fermentation is completed in the cytoplasm. There's no mitochondria. It doesn't take place in mitochondria. And it only gives 2 ATP per glucose molecule. Know that for the test. In fermentation, how many ATP do you get? 2. Where does it take place? In the cytoplasm. For aerobic respiration, we get 36 ATP per sugar molecule. And where does it take place? In the mitochondria. So 36 ATP, know that it takes place in the mitochondria. All right, so here is aerobic aerobic respiration with oxygen. It's not fermentation. So it starts in the cytoplasm of the cell with glycolysis and then moves into the mitochondria for acetyl-CoA Krebs cycle and for the electron transfer phosphorylation. All right. So there's your take-home message, just a review of what we just covered. All right, so let's talk about glycolysis, where it all starts. The reactions of glycolysis convert 1 molecule of glucose into 2 molecules pyruvate for just a net yield of 2 ATP. An energy investment of ATP is required to start glycolysis, to have activation energy to get this thing going. So 2 ATP are used to split glucose and form 2 pGals. There we got our pGals again from last chapter, each with 1 phosphate group. Enzymes convert 2 pGals to 2 PGAs, forming 2 NADHs. 4 ATP are formed by substrate level phosphorylation, which gives us a net of 2 ATP. Glycolysis ends with the formation of 2 3-carbon pyruvate molecules. That doesn't mean much, does it? Let's get to the picture. The picture helps us greatly. All right, so here's our glucose molecule. What's the first thing that happens? You gotta add a phosphorus to it. Do you remember when we talked about enzymes and we talked about there was an enzyme, hexokinase? Remember we saw the pictures of how it has that active site and glucose fits in and the tail of it sticks out and it's able to grab that phosphorus and add it to the glucose. That's this. This is the first stage of glycolysis. So this is hexokinase right here that adds this phosphorus. It takes an ATP molecule. So it takes a phosphorus from ATP, so now he's back to ADP. He doesn't have the energy, so it's going to go back and get some more energy somewhere. But now we have this. Now, what is really neat about this? Remember diffusion, when we talked about, an area of greater concentration wants to go to an area of lesser concentration. Well, your cells want to store a lot of glucose in there for energy that it's going to need. But guess what's going to happen by diffusion? That glucose is going to want to just go back out. So what happens here is, or it's going to stop, glucose is going to stop flowing in. As soon as it adds that phosphorus to it there, it's no longer glucose. So now more glucose are going to keep coming in. And this molecule now can't go out the passive channel. So now it's trapped inside of there, but it's no longer glucose. So more glucose will keep flowing in. Just a little tidbit there for you, just so you know how your cells keep that level of sugar inside of it. All right, now we use another ATP to put a phosphate on the other end of it, and there we go. Now we have the glucose molecule with the phosphate on either end of it. Immediately, that will break apart into 2 3-carbon chains that each have 1 phosphorus. All right, so now, so enzymes attach the phosphorus to the 2 pGals and transfer 2 electrons and a hydrogen ion from each pGal to NAD plus, giving us 2 PGAs and 2 NADHs. So at this point, NADs are going to come and take a hydrogen and form an NADH. So as the phosphoruses are attached here, then you'll notice we have 2 phosphorus now added. So we had 2 phosphorus over here with the 2 NADs. That energy is going to attach a phosphorus, but also attach the hydrogen to the NAD. So now we have 2 NADHs. So far we've spent 2 ATP, but we have gotten out of it so far 2 NADHs. So now a phosphate is taken off each one of those and added to an ADP, so we form two ATPs. And so now we have two ATP formed, and now we take the other phosphorus off and we form two more, and we end up with two pyruvates. That's glycolysis. It takes place in the cytoplasm of your cells. Just about every cell, glycolysis is taking place. To take a sugar and you spend two ATP to get the reaction started by adding phosphoruses onto either side of it. Then it forms two NADHs by adding another phosphorus with NAD, and that hydrogen ion is attached to it. So a hydrogen ion is taken off, a phosphorus is added here, hydrogen goes to NAD, the phosphorus here comes from phosphorus in the cell, and then as you take those phosphoruses off, you're going to form ATP. So what's the product? We have two pyruvates, and we have a net gain of two ATP. We spent two, we get out 4 net gain. That's it. So that's glycolysis. So what have we done basically in glycolysis? We broke sugar in half, but we got a net gain of two ATP and two NADHs. All right, so there's our take-home message. Now, the second stage takes place in the mitochondria, and it completes the breakdown of glucose that began glycolysis. It includes two sets of reactions, acetyl-CoA formation and the Krebs cycle. Each occurs twice. Why does each occur twice? Remember, for one sugar, we get two pyruvates, and it's pyruvates that run through the second stage. So for every sugar, we're going to do this twice because we got 2 3 carbons instead of 1 6 carbon chain. All right, the inner compartment of the mitochondria, enzymes split pyruvate, forming acetyl-CoA, and carbon dioxide diffuses out of the cell, and NADH is formed. There we got another NADH formed. The Krebs cycle is a sequence of enzyme-mediated reactions that break down the acetyl-CoA formed and the acetyl-CoA part into 2 carbon dioxides. Then we have oxaloacetate is used and regenerated. So 3 NADHs are formed and 1 FADH2 and 1 ATP is formed. So again, that doesn't mean a whole lot to you, I don't think. Let's look at the picture and I'll tell you how this works and what you'll need to know for the test. So here is the acetyl-CoA and the Krebs cycle. Excuse me. All right. So here's our pyruvates. We get 2 of these from glycolysis. So what's going to happen first? There's a coenzyme A. That's where we get CoA here. And it's going to take one of those carbons, rip it off, and form an NADH. All right, so now what's happened? Well, we formed another NADH, but notice here, carbon is released. That carbon is put together with an oxygen, and it forms carbon dioxide, and that's gone. So one of the carbons of the 3 is gone. But now it's a 2-carbon chain called acetyl-CoA. Now coenzyme A again now will come through here, and here is oxaloacetate. There's 4 carbons, right? It adds the acetyl-CoA to it. So how many are there? 1, 2, 3, 4, 5, 6. 4 plus 2 equals 6. We're back to a 6-carbon chain. But now what's going to happen? Here we have one of those carbons taken off. It's releases carbon dioxide, and we form an NADH. So remember up here, there was 3 carbons. 1's gone. A carbon dioxide forms. Now there's 2 left. Another one is taken off, and NADH forms. Carbon dioxide is released. And here the 3rd one is gone. And it forms another NAD. So every time a carbon dioxide, every time a carbon is taken off that chain and a carbon dioxide forms, it's going to form an NADH. But it's going to release also carbon dioxide. So now, at this point, all 3 of the carbons that was in pyruvate, gone. They're now part of carbon dioxide, and they've left. We breathe out carbon dioxide. That's where the carbon dioxide that we're breathing out comes from, because we're breaking down sugars and releasing these carbons here in the acetyl-CoA and Krebs cycle. Now, So now we're back down to a 4-carbon chain, right? So we had a 4-carbon, we added two to it here to make 6, we've removed 2, so all 3 of those are gone, we're back to a 4-carbon chain. Now all that's going to happen is we're going to recycle that 4-carbon chain to get back to the oxaloacetate. So doing that, we're going to make an ATP, so we're going to take a phosphorus off and add it to ADP, and we're going to form 2 more coenzymes, FADH2 and another NADH. So now we're back to oxaloacetate. So now this cycle will just start again, because remember, that's just 1 pyruvate. From every glucose, we get 2 pyruvates. So this is going to happen twice for each. So how many, from 1 sugar, how many NADHs is going to form here? 1, 2, 3, 4 times 2. So now we got 8 NADHs from 1 sugar. How many FADHs are going to be formed? Well, there's just 1, so 2, because each pyruvate will go through. How many ATP are going to be formed? 2. But what's important are these coenzymes. We got, look at that, 1, 2, 3, 4, 8 NADHs made, 2 FADH2s. So that's going to play a big role here in the last part. So again, so here is the pyruvate. Coenzyme A will take a carbon off, it releases carbon dioxide, and it forms an NADH. Here, coenzyme again takes acetyl-CoA, it forms acetyl-CoA. It adds acetyl-CoA to citrate, and by the way, this is now citric acid, this right here, citrate. When I was in school, this was called the citric acid cycle. Well, they've named it to the Krebs cycles now. So the Krebs cycle starts as one of the carbon atoms is transferred from acetyl-CoA to oxaloacetate, citrate forms, the coenzyme is regenerated. And now a carbon is removed from the intermediate and leaves the cell as CO2, NADH again. So this is everything that we just covered. I like this. Pyruvate's 3 carbon atoms have now exited the cell as carbon dioxide. And then 1 ATP forms, NAD combines with hydrogen to form FADH2, and now we have an NADH form, and we're back to oxaloacetate, and we start the whole process again. All right, so what do we get out of it? 2 ATPs, 8 NADHs, 2 FADH2s. Right now, there's still not much energy coming out of this sugar. Remember, we got 2 ATP net gain from glycolysis. All right, so that's a little bit. And we get 2 out of this, acetyl-CoA Krebs cycle. Still not much energy out of a sugar molecule. All right, so the second stage of aerobic respiration, acetyl-CoA formation and Krebs cycle occurs in the inner compartments of the mitochondria. So we move in the mitochondria. The pyruvate that formed the glycolysis is converted to acetyl-CoA and carbon dioxide. Acetyl-CoA enters the Krebs cycle, which breaks it down to carbon dioxide. For 2 pyruvate molecules broken down in the second stage reactions, 2 ATP form, 10 coenzymes, 8 NAD, 2 FADH2, and are reduced. All right, how about the big energy payoff? So many ATP are formed during the 3rd and final stage of aerobic respiration, the electron transfer phosphorylation. It occurs in the mitochondria, results in attachment of a phosphate to ADP to form ATP, and that's going to happen exactly the same way as it did in the light-dependent reaction with the enzyme ATP synthase. So the coenzymes, NADH and FADH2, that were made in the Krebs cycle, donate electrons and hydrogen ions to the electron transfer chains. Active transport forms a hydrogen ion concentration gradient in the outer mitochondrial compartment. The hydrogen ions follow its gradient through the ATP synthase, which attaches a phosphate to ADP. Finally, oxygen accepts electrons and combines with hydrogen ions, forming water. And this is critical too. All right, here's the picture. This should look familiar. Remember in the light-dependent reaction, we had photosystem 2 here and photosystem 1. Well, we're getting rid of one of those systems. All right, so now instead, let's call this photosystem 2. It's here, but instead of absorbing energy from sunlight, it's going to take an energy as this hydrogen bond breaks off. So it's going to take that energy, so now that electron's all excited, and it's also formed a hydrogen ion. That electron's so excited, this is the stairway, this is the electron transfer chain. Every time he goes down one of these steps, he's releasing a little bit of energy, and there's an enzyme here that's going to grab hydrogen and pull it through this membrane system. It's an active transport protein, but it needs energy. Where's it getting the energy? From this excited electron here. So now, remember what happened in the light-dependent reaction? There's a whole bunch of hydrogen ions inside, there's very few outside. Diffusion says, we want to get out, we can't get out. Wait, there's a door here, we can go out here. This guy is the exact same enzyme. This is ATP synthase. So every time one of those hydrogen ions goes out, remember how I talked about causes it to turn, so it grabs a phosphorus and turns and attaches it to an ADP to form ATP. Now, because of the energy in 8 NADHs and 2 FADH2s, they're able to pull in 32 hydrogen ions. And as those 32 hydrogen ions leave, they will form 32 ATP. All right, here's another breakdown of it because it is a little bit more complex than what we just showed. There's an enzyme that actually takes the hydrogen off of NADH, and now you've got the hydrogen ion, but now the electron goes down. Here's another enzyme that works with FADH2. Here's the enzyme that's pulling in the hydrogen ions with that energy as it goes down the steps, and here's another one. And so they're all in here. They leave through ATP synthase to form ATP. Typically, the breakdown of one glucose yields 36 ATP. Know this for the test. In aerobic respiration, how many ATP are usually formed for every glucose molecule? 36. How many are made in glycolysis? You need to know this. There's only 2. 2 net gain for glycolysis. How many are formed in acetyl-CoA and Krebs cycles? Again, 2. Now we've got a total of 4, but that's still not much. How many are formed in electron transfer phosphorylation? 32. Here is our big energy payoff right there. So here's just a breakdown of it. So we get 2 from glycolysis, we get 2 from the Krebs cycles, 32 from electron transfer phosphorylation. And what's the end result? Oxygen is released in water. Let me go back to these pictures. And this one is, we have an oxygen that comes out here at the end of the chain. It doesn't tell us here. Look what happens here. The oxygen that comes out the end of this chain is attached to hydrogen ions inside and it forms water. So what's the products of breaking down sugar? We've got all this ATP coming out and we have water. So water is formed. So what is the electron acceptor at the end of the electron transfer chain? It's oxygen that will then be used to form a hydrogen, take a hydrogen ion and attach it, take another one and attach it to form water. All right. Again, here is our final products. We got 36 ATP, oxygen released with water. All right. I'm sorry, oxygen goes in, carbon dioxide is released back up here and in the Krebs cycle. So oxygen has to go in, carbon dioxide released. All right. Fermentation. What happens if there's no oxygen? Well, we go through anaerobic respiration, and another word for anaerobic respiration is fermentation. Fermentation pathways break down carbohydrates without using oxygen. The final steps in these pathways generate NAD+, but do not produce ATP. Glycolysis is the first stage of fermentation also. It does form the two pyruvates, and it makes two NADHs and a net gain of 2 ATP. Pyruvate is converted to other molecules, but it's not fully broken down to carbon dioxide and water. Alcohol is made. It regenerates NAD+, but doesn't produce ATP. Provides enough energy for some single-celled anaerobic species to exist. Bacteria, archaeans can exist with such little energy payout from sugar. Alcoholic fermentation takes place in plants. Pyruvate is split into acetaldehyde and carbon dioxide. Acetaldehyde receives electrons and hydrogen from NADH, forming NAD+, and here's our product, ethanol. This is the basis of brewing. This is moonshine. This is forming alcohol. In animals, we form lactic acid or lactate. The pyruvate receives electrons and hydrogen from NADH, forming NAD+, and lactate, lactic acid, which is not a good deal. All right, so here is alcoholic fermentation. This is glycolysis. We got a glucose, and it's made into two pyruvates. What's the net gain? 2 ATPs. Now, there's no ATP formed after that. That's it, just 2. But the NADHs that were made are now going to be used to change the pyruvates into ethanol. So we're going to lose the NADH formation, but that's going to make ethanol. So what's the net gain? 2 ATP and alcohol. That's it. That's the end of fermentation. But it's the basis of our brewing industry. It's the basis of our baking industry. This is gluten fibers in bread that have captured carbon dioxide being released from fermentation. And this is a picture of yeast cells. Some of these are budding through asexual reproduction, but they break down sugars without oxygen and cause fermentation. All right, so here is the lactic fermentation. This happens in us and the animals. It's still the same up here. Glycolysis, okay, that's good. What's the net gain? 2 ATP. We made 2 NADHs, but those 2 NADHs, instead of working to form these pyruvates into ethyl alcohol, ethanol, we form lactic acid. So that's why when you're running, you start to get that burn in your side. That's a lactic acid buildup, so it's starting to burn. It means you're not getting enough oxygen. You need to breathe better. If you're in sports, they should be working with you about, especially like basketball players who run just so much, to make sure you have breathing techniques to keep a good flow of oxygen in, so you don't get that buildup of lactic acid. All right, red and white muscle fibers. Red muscle fibers make ATP by aerobic respiration, oxygen. Why are they so red? Well, there's a lot of mitochondria, but that's really not what makes them red. Here it is. Myoglobin is in those cells that stores oxygen for sustained, prolonged activity. White muscle fibers, on the other hand, make ATP by lactate fermentation, so you don't get much out of it, but they have few mitochondria, no myoglobin, they sustain short bursts of activity. Now, you're used to these red and white muscle fibers. At Thanksgiving, what do you like to eat? Do you like to eat the dark meat or the white meat? I like the white meat a lot better myself. Some people prefer the dark meat. I like the white meat. My wife likes the dark meat. She likes the eyes, and I like the wings and the breast. Same way with chicken, right? Now think about that for a minute. Can chickens and turkeys fly? Yeah, but just for short little periods, right? So the breast, which controls the wings, and the wing muscles themselves are white muscle fibers. They are good for just short bursts of energy. That's why when a chicken starts to fly, it's gonna run out of oxygen real quick, and it's gonna start building up lactic acid, and so it can't do it prolonged. It's good for short bursts of energy. So then it comes back down. How does a chicken get around? 99% of the time, it walks. So it needs those muscles to be continually walking, and they can run pretty fast. Trust me, I've tried to chase down a chicken before. So the legs, the thighs are darker, that red muscle fibers, for long, prolonged activities. So think about this. If you're a sprinter, guess which type of muscle you're going to have more of in your system. If you're better at sprinting than you are marathons, you're going to have more white muscle fiber. If you're better at marathons than you are sprinting, you're just not that fast sprinting, but man, you can go forever, you got a lot more red muscle fibers. So that's how it breaks down with us. So here is just, this is a slice of muscle, and you can see there's some dark muscle cells and some light. A lot of times there's a mixture of them because sometimes you do need quick bursts so your muscles can work quickly. Sometimes you need that slow, steady wins the race. Guess what kind of muscle your heart is? It is just about all dark. Why? Because it's got to keep going constantly. So, a lot for prolonged duration. So it'll be a dark muscle fiber. All right, so what's fermentation? We got that. Now, alternate energy sources in food. Where are you going to get most of your energy from? Simple sugars as you eat. But most of your diet should not be simple sugars. Only about 10% of your diet should be simple sugars. What happens if you eat too much simple sugars? Blood sugar goes up, your body's going to start grabbing down your blood and forming fat. So, there you go. So, your body will store that as fat. So, that is for energy storage. Wonderful. So, your body can break down simple sugars. Great. It can break down fats by converting them to simple sugars and also acetyl-CoA, which will go into the Krebs cycle. So, that's good. Your body can also break down proteins. That's not good. If you're starting to break down proteins, that's a little bit much. You know, as you eat proteins, you get too many amino acids, and so your body has too many amino acids in there. So yeah, you do need to break those down. And you can actually break apart amino acids and plug them in different, depends on the amino acid, of where you plug it in to the glycolysis or to the Krebs cycle or to acetyl-CoA formation. Depends on the amino acid. But if you're in a state where you haven't eaten anything, yeah, your first thing you're going to break down is simple sugars. The second thing you're going to break down is fat, which is good. You know, that's why we exercise, to try to keep your fat level down. But once you have broken down all your simple sugars, all the complex carbohydrates are gone, the fats are gone. When you get to the point where you're breaking down protein in your body, that is not a good thing. Because proteins are critical for things to function. And so, you know, it's the last line of defense. Now, you do break apart proteins as you eat them, don't get me wrong. It's good to break down proteins there. But like for animals who hunt and they can't find new prey, you know, they will gorge themselves to form a lot of fat. They will use that fat. Remember bears in hibernation, they gorge themselves to form all that fat. They go in hibernation and then they use all that fat for energy while they're in hibernation. If they stay in hibernation too long, or when they come out, they can't find food, then they can start breaking down protein. And when you start breaking down protein, you're starting to break down some muscle and some different things. So that is not great. So enzymes break starch and other complex carbohydrates down to monosaccharides. That's great for quick energy. Monosaccharides are taken up in cells and converted to glucose 6-phosphate, which continues through glycolysis. So that's the normal cycle we just studied. A high concentration of ATP causes glucose 6-phosphate to be diverted from glycolysis into a pathway that forms glycogen, and this is the first step in forming fat. You get that glycogen glycerol head, attached fatty acid tails to it, and there you go. You got energy stored, which is good. We need to be able to store energy. So here's starch. That's a complex carbohydrate, and what does digestion do? Takes each of these glucose molecules off. We can burn that as energy. So this is how plants store excess sugar. If we break these down, eat a lot of carbohydrates, and you've got way too much glucose molecules floating around your system, then if you break it down, your body's gonna start taking those and forming glycogen. All right, so enzymes cleave fat into glycerol and fatty acid tails here. Glycerol produced products enter glycolysis. So when we start to break down fats, whenever you run out of sugars, the glycerol head is broken down, that enters glycolysis. The fatty acid tails are converted to acetyl-CoA, and they enter the Krebs cycle. So going through these cycles to produce ATP or produce those coenzymes that can go into electron transfer phosphorylation. Fatty acid breakdown yields more ATP per carbon atom than carbohydrates. So there's more energy stored in fat that's even in regular sugars. When blood glucose level is high, acetyl-CoA is diverted from the Krebs cycle and into a pathway that makes the fatty acids. So there you go. Too much sugar is a bad thing. You'll form fat. Not enough sugar, you're going to break down fat and so it'll enter glycolysis and acetyl-CoA formation. So there's a triglyceride. There's that glycerol head. Here's the 3 fatty acid tails. Remember there's double bonds there to cancel them out. So this is not a saturated fat. That's an unsaturated fat. All right, enzymes split dietary proteins. Here we go. This is, if you have to get energy from proteins, which is a normal part of our diet. If you eat eggs and lots of meat, you're going to get way too many amino acids in your meat. So you can start digesting those excess amino acids. So you can split proteins into their amino acid subunits, which are used to build proteins, other molecules. The amino group is removed and converted into ammonia. So if you need to break it down, you need to get rid of it. Part of it goes to ammonia, which is a waste product. You eliminate that in your urine. Acetyl-CoA can then be formed from the rest of it. Or, depends on the amino acid again. Some amino acids you can form a pyruvate. So you take out ammonia. Some you form acetyl-CoA, go into the Krebs cycle. Some you form pyruvate. You go through acetyl-CoA and the Krebs cycle and electron phosphorylation, or an intermediate of the Krebs cycle forms. You can even form from amino acids, p-gals. So depending on what the acid is. So there is an amino acid, and that can actually be the nitrogen taken off of it, and it forms a pyruvate, and now it's ready to go through acetyl-CoA, Krebs cycle, electron transfer, phosphorylation. Here's just a breakdown of that. So here's our food of what we're eating. Part of it should be simple sugars. That goes directly through. Part of it should be complex carbohydrates. We break that down into simple sugars. It goes through. If we get too much simple sugars, what's going to happen? It's going to store it as fats. Then the fat can be taken. The glycerol is taken and formed into the glycerol can be taken and run through glycolysis. The fatty acid can be taken here through acetyl-CoA, and then proteins, depending on which it is. Some of them can enter as pyruvate, some of them enter as acetyl-CoA, and some of them enter as intermediates of the Krebs cycle. But we get the energy out of it. So you can break down sugars, complex carbohydrates, fats, and proteins to get energy. All right, there's points to ponder as you think about this. I added this to the end of this. I wanted you to see this. I know you can't see the details of it, but this is your metabolic activity of one cell. This is all the different chemical reactions taking place at all times in every cell. And if I remember correctly, which one is it? It's one of these two. I think it's this one is the Krebs cycle as we break down sugars. And that's right there. But look at all the activity that's going on in your cell at all times. And every one of these reactions take energy. Where are you going to get the energy from? Breaking down sugars to make ATP. 36 for every glucose with there's oxygen present in the mitochondria. So that is the end of chapter 7. All right, that's the second test will be chapters 5, 6, and 7. So we're ready now at this point to start thinking about test number 2. I'm not yet. I'm still working on test number one. We're actually, this is Thursday of the first week that I'm taping this. So working tonight, I will finish test number one. I'm going to get that loaded into Moodle. We will take test number one on Tuesday, but you need to go through that Proctor Track onboarding quiz. Don't worry about that quiz. It's the stupidest question I could come up with. What color is an orange is your quiz question. You actually get a point for that, so there you go. But it's to get you set up so that you can take the test. So we'll be looking at all that stuff as we go along here. Probably when you watch this video, you've already taken that first test. I hope things work out. So we will see how that works. I hope things work nicely for that. If you have any problems, don't hesitate to text. Let me give you a heads up too. By this point, you're probably thinking, what did I make on that first test? You can go look at it in Moodle in the grade book. It's going to tell you your score, but don't go by Moodle grade book. I will curve your tests. That's going to tell you your raw score. When you look at Moodle, don't think that this is your final grade. I'm going to help you as much as I legally can. I will curve some of these tests because some of this information is pretty tough. I will curve your tests, and we're going to add those papers to it, which will be weighed differently. So as we go through the semester, if you're curious about your grade, just text me, and I can figure up at that point what your real grade is. I will probably send you out individual emails after each test to tell you what your final grade is too. So if at this point you're starting to wonder, what did I make on my test, start checking your email. I will send you some information to let you know what you made. So thanks, and I'm going to add, before the week is out, week number 2 is out, I'm going to add a review so to help you get ready for test number 2. So we'll see you soon.